So let's discuss passive transport. So the passive transport includes three types of transport. One is simple diffusion, facilitated diffusion, and osmosis. So simple diffusion. Here's your bilayer, right? One side is hydrophilic and internally inside it's hydrophobic. So it's easier for the lipid molecules because they are hydrophobic, and most of the membrane is hydrophobic, made of fatty acids. So for the fatty acids, for the lipids, it's easier to pass through the membrane without a transporter. These proteins, transmembrane proteins, are the transporters. So the lipids can be diffused from high concentration. You see high concentration, meaning there's more molecules, and low concentration, there's less molecules. So it's easier to go from high to low concentration, right? So you have a full room of people and the next room is empty. You don't want to stay in this room because it's hot and there's a lot of people. So you want to go to where there's less of people. So you're going from high concentration to low concentration gradient. And the concentration gradient, that's your membrane. So it's called simple diffusion. You just diffuse simply without the presence of a carrier. Then there's a carrier-mediated, facilitated. So this is the mediator, carrier-mediator, and this is also a facilitator. So it facilitates of a molecule if the molecule will fit the shape. You see the diamond shape of a molecule, and there's a diamond shape of this carrier. But it's still part of the diffusion, simple diffusion, facilitated diffusion, but it's part of the passive transport. You also go from high concentration to low concentration. In this case, it doesn't matter if it's a lipid molecule, doesn't matter, right? So here the diamond, like let's say sugar. So it also goes from high concentration to low concentration, but with the carrier, right? So the body, the cell controls what can enter. Here it's kind of uncontrollably you can enter, but here with a control. So because here is a facilitated, it's been facilitated like a door. If you can fit the shape, you will be able to get inside. Carrier-mediated facilitated diffusion via protein carrier specific for one chemical because it fits the shape. Binding of solute, this is the solute, causes transport protein to change the shape, right? Changes the shape, so you enter, then it opens and you enter, but still going from high to low concentration because here is your gradient. On this side you have more, on this side you have less. You're passing, it's still passive transport. Why is it passive? No energy required. From high to low. Then is a chemical-mediated facilitated diffusion, right? I'm sorry, here's a channel, not chemical. Chemical, there's also chemical. So there's a channel-mediated diffusion. Here's the carrier, it carries, fits the shape. And here is just a channel. They're a similar one to another. But you're also going from high concentration gradient to low concentration gradient through a channel-mediated protein. Facilitated diffusion, facilitated diffusion. So here, facilitated and facilitated is the key term. Facilitated. So every time you hear facilitated, you should have a carrier. Through a channel protein, mostly ions, right? Mostly ions, positive or negative, that's the ions. You need to control them, but still going from high concentration to low concentration. Let's say sodium. There's more sodium outside of the cell, more, and there's less sodium inside of the cell, less. So let's say there's lots of sodium molecules, that's what they represent, arrow, more, and here's the less sodium. So you go from high to low through the carrier, through this channel. And here is the water going through the osmosis, through aquaporins, aquaporins, But the water goes from high to low, right? So if there's more water in this room, you open the door, there's no water, so the water will diffuse from high to low, right? Understandable. But it also going through that carrier, right? Through this channel, from high to low. So the osmosis is the movement of water from high concentration to low concentration. It is all passive transport. But I want to add something to osmosis. So the osmosis, and please understand that and memorize that. That would be very important. Osmosis, movement of water from high of water to low of water. Understandable, passive transport. But here is your gradient. If on this side you have solutes, let's say sodium, and on this side of the membrane you have solutes, sodium, there's a higher concentration gradient of a solute. Here's a low concentration gradient of a solute. Water will always go towards high concentration gradient of a solute. I will be also explaining this in class because this is a complex and mind-boggling thing to discuss. But for now, just state that water moves from high to low concentration of itself of water. But when the solutes are present, and there's a semi-permeable membrane that can only permit movement of water, but molecules cannot fit, let's say the big molecules, and the channels cannot permit molecule to fit through. So the water will go from high to low concentration of solute. Diffusion from high concentration to low concentration. Ions, or as you've seen the other molecules, right? We're not talking about osmosis for now, just the diffusion. Concentration. Greater the difference of concentration before two areas, the faster the diffusion. Meaning if you have greater difference, here higher concentration of the molecules, here's the low concentration of the molecules. So you want to go there, right? So this is lots of people in the room, and it's too hot. There's a lot of people. You want to escape that environment. Same thing with molecules. And molecules within the environment, they move fast, so they want to go to where there's less concentration. And when they go to where there's less concentration, because that membrane can permit to move them, they will disperse equally. So you have more here, have less here, but you start moving them, and now you might have the equal amount on both sides, so you disperse equally. So the concentration, the greater the difference of concentration between two areas, the faster the diffusion occurs without energy. We're only talking now without energy. Molecular size. The smaller the molecule, the faster it will pass through. Temperature. Higher temperature increases kinetic energy, which results in the faster diffusion. Higher temperature, smaller molecules, higher concentration of solids. All of them will result in the increased movement or dispersion of the molecules between two areas. Equilibrium, it will reach the equilibrium from high concentration to low concentration, and now you reach the equilibrium when there's no net movement, right? Now you have equal amount of molecules on both sides, or solids. And the molecules that we're going to use in example are sodium, chloride, potassium. Definitely those three. So that's all passive transport. No energy required. Passive transport. Molecules have the natural drive to diffuse down concentration gradient that exists between extracellular and intracellular areas. Here's the cell. This is the extracellular space. This is the intracellular space inside the cell and outside of the cell. So there's more sodium outside of the cell and there's less sodium inside of the cell. There's less potassium outside of the cell and there's more potassium inside of the cell. So that's why it's easier for the sodium to get inside from high to low, and that's why it's easier, it would be less. And that's why it's easier for potassium to get outside of the cell, because there's more potassium inside and there's less potassium outside. So you go from high to low. And each molecule goes to its own molecule in this example. Non-polar hydrophobic lubricant, also called differential equilibrium. Simple diffusion. Molecules that may passively diffuse to the membrane include lipid soluble and non-polar substances. Facilitated diffusion, larger non-lipid molecules. Osmosis, right? So let's look at osmosis. Osmosis is a special name for movement of solvent. Water, right? Universal solvent. Usually water, not molecules. That's for this example, but we're going to get to something else when it comes to a osmosis. But here's actually a good example of osmosis. So you see, it says here, selectively permeable membrane. Here's a high concentration of solids. You see there's more, 1, 2, 3, 4, 5, 10. On this side is only 5. So the water will move towards highest concentration gradient of a solid. That's why now we have less here of water, because the water moved towards high concentration gradient of solid. Because the membrane is only allow water to pass through the membrane, not the solid. Solid still stays on this side. Freely, here's a selectively permeable, meaning that only water can pass through, but it takes out the molecules, so the molecules cannot pass through. So that's why water goes towards highest concentration gradient of a solute. And here, freely permeable membrane, meaning that solid can pass through. You see it passes through, right? This red circle can pass through the membrane. So when it can pass through the membrane, that all the solids goes here. So they disperse on the other side of the cell. So now they equally disperse and water disperses as well. But here water goes towards high concentration gradient of a solid. When it comes to osmosis, that's a concept that you have to understand, but I will be discussing that even more. So simple diffusion, right? From high to low. Things like oxygen, carbon dioxide, steroid hormones, fatty acids, they do not need any carriers. They go from high concentration to low concentration. Carbon dioxide, it's nonpolar. Steroidal hormones, it's a lipid. Fatty acids, gas, it's easier for gas to get inside from high concentration to low concentration. Facilitated diffusion, so here's your carrier-mediated, the protein, so the fits tape, it passes through. That's transmembrane integral protein that spans through the membrane. So things like glucose, amino acids, some ions, like in this example, can pass through this channel-mediated proteins, also going from high to low, no energy required. Leakage channels, gated channels, we'll get to this, binding limited number of carriers, and active transport. So up to that, we just discussed the passive transport that does not require energy. So with the passive transport, osmosis, and facilitated diffusion, right? But active transport requires energy. So the difference between passive transport and active transport is that passive transport, high concentration, you go to low concentration. Passively, without mediated channels, or with facilitated mediated channels, right? Or through aquaporins from high to low. But active transport, you are able to go from low to high concentration. You see the passive transport, you go from high to low, but active transport, able to go against concentration gradient. You go from low to high. Great example would be standing on the platform. So the train arrives to the platform, doors are open, and the car is packed. So you're not able to get inside of the car. You need an energy, someone pushing you inside of that car, train, inside of that, so the door can close behind you. So you need the energy. In order to go against concentration, pretend there's like lots of people here and like up to the door. So you need someone from the outside, from the corridor, start pushing you inside. That's an active transport, active. When you hear active, energy going against concentration gradient, inside. So the passive transport from high to low, regardless if there is a channel, but the active transport from low to high concentration. Here's from high to low concentration. I will like that, right? But also there is a channel. Here's lots of examples on that. You can read all this, but this would be the most important thing to understand that active transport. You go from low to high concentration gradient with the help of energy. And the sodium-potassium pump is a great example. We will discuss this further down in the nervous system. So for now, just get this, that. We will be getting to that in the nervous system. I don't want to pile everything into one lecture.